Biology 107 Lab 6, Glucose Metabolism in Yeast. In today's lab, we will be looking at fermentation, a form of carbohydrate catabolism. Specifically, we will measure glucose fermentation in yeast, a single-celled eukaryotic organism. Let's take a quick look at glucose catabolism. Here is our glucose molecule, and here is our yeast cell. First, the glucose moves into the cytoplasm, and then the breakdown of glucose begins with the reactions of glycolysis, where one glucose molecule is converted into two pyruvate molecules. Glycolysis also produces two ATP molecules, and here is a summary of glycolysis. We have two ATP produced, two NADH produced, not shown, and the last thing to note is that glycolysis occurs in the cytoplasm whether oxygen is present or not. In most organisms, when oxygen is present, aerobic respiration occurs. In aerobic respiration, pyruvate is moved into the mitochondria where it is converted to a molecule called acetyl-CoA. After this, it is eventually converted into carbon dioxide through the reactions of the citric acid cycle. Here, another two molecules of ATP are made, which is good because cells like ATP. After this, a lot more ATP is made during the process of oxidative phosphorylation. This includes the electron transport chain and chemiosmosis. We will talk about those a lot more in lecture in the coming week or two. Without oxygen, pyruvate is only partially broken down using fermentation pathways. The two most common are alcohol fermentation and lactic acid fermentation. So one way to look at this diagram is to realize that pyruvate is the branching point. All cells convert glucose into pyruvate, and if oxygen is present, it will get broken down further and produce a lot of ATP in the mitochondria. If no oxygen is present, the pyruvate will only get broken down partially into fermentation pathways. Yeast contain the enzymes to undergo alcohol fermentation, producing ethanol and carbon dioxide. During this process, pyruvate is first broken down into acetaldehyde and carbon dioxide. Then the acetaldehyde is broken down into ethanol. The last step plays a key role in regenerating NAD+, a key component required for the continuation of the glycolysis pathway. We are going to talk about NADH and NAD+ a lot more in our lectures. They are involved in electron carrying and bringing those electrons to the electron transport chain in the mitochondria. Without a source of NAD+, glycolysis would cease entirely, and ATP production would cease entirely, and the cell would eventually die. Interestingly, yeast use this pathway to degrade glucose even when oxygen is present. This is particularly useful for these two products, carbon dioxide and ethanol. Carbon dioxide allows dough to rise in bread making, and ethanol is a product important to the alcohol beverage industry and also is used as a biofuel. In today's exercise, we will be conducting a fermentation reaction using different parts of the corn plant as a substrate: the stem, the kernel, and the cob. There is a lot of interest today in finding renewable energy sources, and corn is an ideal crop for that. We can grow a lot of corn very quickly in per acre, per hectare, and it produces a lot of starch, and starch is actually a really good thing for biofuels because it's very easily converted to ethanol. Ethanol can be used in a lot of processes, particularly we like to supplement our gasoline with ethanol as a biofuel. Ideally, if we could use the entire corn plant, we would be extremely efficient in producing ethanol as a biofuel. That's what today's experiment is all about. Here's the setup for today's experiment. You can see we have 6 tubes, each of them containing a different substrate that's going to be used by the yeast for glucose fermentation. The first 2 are basically controls. You can see we have a water control, which is a negative control, meaning that there should be no glucose in the water and nothing should happen. We also have a positive control made of corn syrup. Corn syrup is highly refined and processed and has a really high concentration of glucose and fructose, so that one should work really well. And then we have our 3 parts of the plant. We have the stem, the kernel, and the cob. So each of these were basically processed by throwing them in a blender for a while with a little bit of water and a little bit of buffer until we get a nice good mixture, and then filtered out to get all the particulates. So hopefully lots of glucose and starch in each of those for the yeast to ferment. I'll come to tube 6 in a moment. There's our extracts. You can see the water and the corn syrup are looking just clear, but you can see the stem and the kernel and the cob extracts have different colors reflecting different components in those parts of the plant. So let's talk about tube number 6. Tube number 6, we're going to try one more thing to see if we can enhance our fermentation of the cob extract. The cob extract, we are going to also add in tube number 6 a little bit of an enzyme called amylase. Alpha amylase is an enzyme, and this enzyme breaks down alpha glycosidic linkages. Alpha glycosidic linkages are found all over the place in starch and glycogen and other carbohydrates. And so the idea here is if there's any starch found in the cob, we can break it down into glucose and maltose and other small monosaccharides and hopefully enhance our fermentation process. Back to our experimental setup, I'll show you some photographs of the experiment that I did. So the first step is to make those mixtures, so 5 mils of each substrate except for tube 6, which had 4.5 mils of the cob extract plus 0.5 mils of the amylase. So there they are in the 6 test tubes. The next step in the experiment is to warm up the substrates to 30 degrees Celsius. So there it is in the incubator. The reason why we put it in 30 degrees Celsius is to get the substrate at a nice temperature so the yeast will ferment it most efficiently. There's the yeast suspension. You can see it's 0.2 grams per milliliters. The next step is, after 10 minutes of pre-incubation, I'm going to add the yeast to these mixtures. Here's our experimental apparatus. In each test tube, we will have a substrate and yeast mixture. So this could be yeast and water, or yeast and corn syrup, or yeast and kernel, and so on. In it, we will also have a Durham tube. A Durham tube is basically a smaller inverted tube full of the mixture. You can see on the right the photograph of our Durham tubes. Our Durham tubes have little graduations on them, which is very nice so that we can measure any gas that is produced. Fermentation will produce carbon dioxide. So when carbon dioxide is produced, we can measure the volume of the carbon dioxide produced by the reaction over time. Once our apparatuses are set up, we're going to put them back in the 30 degrees Celsius incubator and measure for carbon dioxide gas produced over time. For example, here is the corn syrup at 0 minutes. So you can see at 0 minutes, no carbon dioxide gas has been produced. The yeast and corn syrup mixture goes all the way to the top of the Durham tube. Let's take a look at the 5-minute mark. We can see a little bit of carbon dioxide gas is produced in the top of the tube, approximately 0.05 milliliters. So we're going to do this, take readings every 5 minutes for all 6 tubes until we reach the 45-minute mark. After all that work, here is our data. You can see the water, which is the negative control, turned out really well. It's 0, 0, 0 all the way across, so that means that the water did not ferment. There was no glucose in it, so that's a good sign. You can see all the other samples have varying degrees of success in terms of how much carbon dioxide gas was produced. So now what we want to do is analyze this data, and you're going to do this by producing some graphs. So let's talk about how that's going to work. For our graphs, we're going to put the volume of carbon dioxide produced on the y-axis and time on the x-axis up to 45 minutes. We can throw in our water values. It's all going to be 0, 0, 0 all the way across. And what we can do is connect the dots. In this case here, we don't necessarily have to look for a line of best fit because it's all flat, pretty easy one to do. We'll put another one on the graph here, and this one is not going to be flat. This doesn't represent our data necessarily. This is from a different semester, but you'll get the idea of the shape of most of the other curves. So there's another plot. Most of the other curves will start off relatively slow. They may not produce carbon dioxide in the first 5, 10, or in some cases even 15 minutes, but then eventually they start producing carbon dioxide and you get into an exponential phase. Eventually, at some point, maybe around the 30 or 40 minute mark, it starts slowing down and it plateaus at the top. So we have a little bit of a sigmoidal or an S-shaped curve. Like before, you can connect the dots or you can find a curved line of best fit. It doesn't really matter, but do draw a line so that you can see the different data points and the different lines really well. So what you're going to do is put all 6 of your plots on one graph. And I have a small Excel tutorial to show you how to do this in a moment, and hopefully that will help you to produce your graph. The next step in our data analysis is to calculate the slope of each of our graphs. So what I want you to do is calculate the slope of the linear exponential portion of the curve. So what does this mean? It means we're going to ignore the flat part at the beginning and the flat part at the end and just get that exponential part in the middle. So in this particular graph, what I'm going to do is select that middle portion and I'm going to select the time points at 15 and 30 minutes. I'm going to calculate my slope for that particular range there, and the slope is equal to rise over run. So in this case here, the rise is 0.83 minus 0.18 milliliters, and the run is 30 minutes minus 15 minutes. So my final value for my slope is 0.043 mils per minute. So this means that for this particular treatment, we're making 0.043 milliliters of carbon dioxide per minute. So you're going to do this for all 6 of your plots, and what I want you to do is put all of them on one graph. Right now I have a little Microsoft Excel tutorial is coming next. It's about 7 minutes. If you're going to make your graphs in Excel, you can watch this tutorial. If not, skip ahead in the video for 7 minutes.
You can see here I have the data. It's all inputted into the spreadsheet and ready to go. So what I'm going to do is first select the data, so B3, and highlight all the way to L9. And it's pretty simple. We go up to Insert. I'm going to go over to the chart area. You can see there's lots of options. I'm going to select this one, which is the scatter chart. I'm going to select one of these options. You can see there's lots of options there. I like the one where it connects the dots. And there's my chart. So you can see it is looking pretty good and it's almost ready to go. It's not quite fitting all of the Biology 107 standards. There's a few things we need to do. First thing we need to do is add axes titles. So what you need to do is click on your chart and then go up to the top and click on Design. So if you go over to the far left now, Add Chart Element, we can add axes titles, which are right here. I'll start off with my horizontal title. So my x-axis is time, and so the units are minutes. I can add my y-axis title. So the title here is going to be volume of carbon dioxide produced, and the units are milliliters. I'm going to do one more thing, the little 2, I'm going to make it a subscript. I go to Home, and I select Font, and find the subscript option to make it a little 2. Next thing I'm going to do, I don't really like my legend being on the bottom, I kind of like my legend on the right. So I'm going to right-click that, Format Legend, and you can see there are some options there, Legend Position. I'm going to click Right. My second note says use a different symbol for each line. So if you look at your graph, you may notice that all of these lines have the same symbol. They're all little circles. So that's a problem because if we're going to print this off in black and white, we won't be able to tell which is which. It's all going to be grayscale. If I'm colorblind or the person looking at your graph is colorblind, they won't be able to tell the difference between red and green, or even sometimes the different hues of blue are a little bit hard to distinguish. So what you want to do to change your symbols is click on the symbol like this, and you can right click and go down to Format Data Series. It should pop up here on the right. We're going to click on that little fill bucket, and we want to go here to marker. If you want to change the color, you can do that. I don't think that's necessary. These colors aren't too bad. And I'm going to go with marker options, built in. And what I'm going to do is select a different symbol. So I will select the square for this one. I'm going to make it a little bit bigger too, so it's easier to see. And there we go. So I'm going to go ahead and change the symbols for all of the lines, and then I'll see what note number 3 says. There's the final product. My graph has those nice little symbols, a different one for each, and a different color, so it's much easier to interpret. My third note is saying, hey, let's put the title below. So Excel has this default where they put the chart title up at the top. So what I'm going to do is just get rid of that. And what you can do is just insert the title either in Excel or you can add it in your Word document and just type it in below. So this is Lab 6. So this will be figure 6-1. And I'm going to write myself a descriptive title. And my title is, of course, going to have the binomial name of the organism. In this case here, the organism is yeast. The binomial name you can look up on the internet, or you can look in the lab manual. It's found in both places. Now what I'm going to do is show you how to calculate the slope of the line. It's really important that you calculate the slope of the exponential portions of the lines. So for calculating your slopes of your lines, it can be easily done in Excel. We'll do the water for starters. The water one should be relatively simple because of course it's just 0, 0, 0, 0 and so on, and of course the slope is 0, but I'll show you how to do that anyway. One thing that you can do is make a copy of your Excel file and do this for each of your slopes, or you can do it all on one graph if you like. It's just going to get really cluttery if you do it all in one graph. So what I'm going to do is actually delete all the data for everything except for water. You can see there's my water curve there at 0. If I click on my curve, if I right click and go to add trend line. So if you go down, scroll down, and at the bottom it says display the equation on the chart. And that's what's done right here. You can see my equation here is y equals 0. I'll make that a little bit bigger so that everyone can see it. And that's the equation on my water line. So now let's try looking at one of the other curves, maybe the corn syrup. Here's my curve for the corn syrup. You can see I still have the water on that curve, but I've deleted all the other data so that I can see the curve really easily. Like I said, it's much easier if I do this for each separate set of data points. A lot less cluttery, a lot easier to see, a lot easier to manage. So as I mentioned, you want to get the data for the exponential portion of the curve. So we want to miss this, we don't want this beginning region where it's kind of flat. We don't want this end region where it's starting to slope off. We want this region here in the middle on the curve. So the easiest thing to do is actually just delete that data. So here we go. I'm going to delete the data from this curve, make a copy so you don't have this deleted permanently, and I'm going to delete those last 3 points as well. So there we go. Now I have the exponential portion of the line. All I need to do now is find the trend line. So just like before, add the trend line, scroll down, display the equation on the chart. And there's my equation right there. So I'm getting the slope and everything. I'll just make that a little bit bigger so that you can see that. There we go. And so for the corn syrup, this is the slope that I'm getting. So hopefully you'll get a similar number in your calculations as well. So now after this, you want to do this with a stem extract, kernel extract, cob extract, and cob extract in amylase, and input the slope values into table 6-3.
Once you've produced all your graphs and you've figured out all the slopes of the linear exponential parts, you're going to input this data into table 6-3, and this here is your final data. So when you're discussing your hypothesis, when you're discussing your conclusions, this is the final data that you're going to talk about. You're not going to talk about how much carbon dioxide has been produced. You're going to talk about the rate of carbon dioxide production. This is the important essential data for this particular experiment. Once you have all of your data and your graphs produced, then you can do the short report. The short report is found at the end of Lab 6. This is worth 15 marks. I think this one is just a little bit shorter than the last one, the Lab 4-5 report. You can see I have questions pertaining to the introduction, the results and the discussion, and the appendix. So for the results, you will have one graph with all 6 plots on your graph. You will also have an analysis of the slopes, and that will be part of the appendix. So do take a look at the lab manual or ask me if you have any questions about how that is going to work. It is outlined how you can make the graphs and how you can analyze the slopes in the Excel tutorial found in this video. Last thing I have for you for today is a little cartoon. You can see this is yeast doing their anaerobic exercises. So I thought that was a little cute one, and it's relevant to the topic.